So in this short video, we're going to look at the difference between an autosomal recessive inherited disease versus an autosomal dominant inherited disease. So in the case of an autosomal recessive, we're going to look here at cystic fibrosis. And with cystic fibrosis, it really comes down to the protein channel that is used in your cell membranes to transport chloride ions. And if you remember proteins are made, if you start way back in DNA, where you have things called codons, and codons are your groups of 3 bases. So like here would be 2 codons. Then you, now each of these codons are going to code for an amino acid. So our hopes is that when we have our DNA sequence, and then it's transcribed into messenger RNA here, and then we read each codon to figure out the chain of amino acids. Each amino acid has an area called an R group that varies. And how these R groups interact with each other determines how proteins fold up. So if you inherit a chromosome that has a normal version of the CFTR protein, what that means is that the DNA or the DNA sequence is codes for the correct pairing or correct sequence of amino acids, so that protein will fold up in the correct order. However, if you inherit a mutation, that means that somewhere in the DNA, like maybe a point mutation that resulted in a missense, and now the sequence of DNA that you have at that locus codes for incorrect amino acids, and now it doesn't fold up correctly. So you may still have a protein in your cell membrane, but it's not the correct shape. So for a person to be homozygous dominant, what that means is both of their chromosomes they inherited code for working or have like working alleles. Both copies code for the correct version of that protein channel. If a person is heterozygous, they are totally fine and healthy and are not affected with cystic fibrosis, because here in the heterozygote, you only need like one correct gene sequence to know how to build that protein channel, and therefore being a heterozygote, you make the correct protein and you do not have cystic fibrosis, you are not affected. Now, it's only when you inherit 2 copies that you don't have a working protein channel, so therefore your DNA doesn't have the correct information, so therefore you cannot transport chloride ions. This messes up osmosis, and then you are affected with cystic fibrosis. So we can actually change, instead of writing CFTR, this is where we write those capital and lowercase letters to represent the different alleles or versions that we inherit. So here a person, when it asks like what would be the phenotype of a person who inherits 2 copies, their phenotype would be they would have or be affected by cystic fibrosis. If what would be the genotype of a person hybrid, that'd be this middle person here. Now they would not have cystic fibrosis because they have that one working protein. And then the cross, if you ask what would you write if they were crossed, this is what you would write down. This would be your hybrid. This would be your person affected with cystic fibrosis. Now, this is different than a disease that is inherited as dominant. So now with this, if a Huntington's disease is inherited as autosomal dominant, what does that mean? Well, if you have 2 copies, you can see here this dominant allele codes for the mRNA that codes for an elongated, like a very long version of this Huntington protein. So, and then that Huntington protein, because it is so long, the neuron will actually like chop it up into smaller pieces. And now those little protein bits accumulate and clump together and become like toxic in the neurons. So if you have 2 alleles, 2 copies of this, you're homozygous dominant. What that means is both your versions of DNA code for very long proteins, Huntington proteins, that'll eventually get chopped up into smaller pieces. And it's these smaller pieces that are causing the person harm. Now, if a person is heterozygous, one copy of their one of their chromosomes codes for that elongated Huntington protein that gets chopped up into smaller pieces. And then their other allele, though, codes for the normal working version. At this point, this is really where a dominantly inherited disease is different than a recessive inherited disease. Because here, what we're seeing, it's the presence of this protein, of that long protein that causes the disease. So by being a heterozygous, you only have to have one copy, one allele that has those directions to make that long protein, and you are affected with Huntington's disease. So here, a homozygous dominant and a heterozygous both are affected with Huntington's because only one allele is required in order to make these toxic proteins. Now, if you are homozygous recessive, you make the normal working version of this protein, and you live a normal life. With Huntington's disease, this is a late onset. So this here doesn't start happening until between 30 and 50 years old. So by this point, they've already had children and possibly passed on these alleles. I also want to use this example to think about how just because something is dominant doesn't mean it's the most common. In this case, being homozygous dominant would actually be deadly. So here's my question for you. What would be the genotype of a person affected with Huntington's disease? That's right. They'd have be either homozygous dominant or heterozygous. Now let's think about this. If an individual has a parent with Huntington's disease, what is the probability of them inheriting that allele? Well, here, it depends on what's the genotype of that parent. Are they homozygous dominant or are they heterozygous? Because both of these genotypes would code for Huntington's. However, during gametes separating in Mendel's law of segregation as we make alleles, in this parent, 100% of their gametes will have that dominant allele for Huntington's, and in this parent, only 50% will have that allele for Huntington's. So then, if an individual has a parent who is not affected with Huntington's disease, but has a grandparent who was, what is the probability of them inheriting it? Through this family line, not really talking about mates. All right, so now we're looking at an individual has a parent. So the individual is here, has a parent who is not affected by Huntington's, but has a grandparent who was. What is the probability of them inheriting it through this family line? Well, we're going to, because Huntington's is rare, we assume that when they mate, they're going to mate with a homozygous recessive, because that is the wild type and the most common. But through this family line, because whoever the affected parent mated with, we can see that this offspring inherited this recessive allele as well as this recessive allele. So now all of this offspring's gametes only have the normal version of this allele available. So there's actually a 0% chance that this person will inherit Huntington's from this parent because the parent does not carry that Huntington's allele. Okay? So the difference, again, between autosomal dominantly inherited diseases is that it's the presence of a protein that actually causes the harm. So even being a heterozygote and having just one copy would actually cause harm and have it be expressed in the phenotype. Whereas in cystic fibrosis as an autosomal, here it's the absence of a protein that is causing you harm. All right, good job.